Welcome to this biochemistry video on DNA replication. So, in cells, DNA in the chromosome has two main functions. First off, it is obviously the information template for producing proteins, but it also replicates itself, and this process is called replication. Replication begins at the point in DNA called the origin of replication, or the replication fork, and this is what you see here. We're going to talk about all of these different proteins, enzymes, and the different features of the DNA. So step zero. Step zero is when we have, so normally DNA is packaged into histones, and there's going to be some kind of event, methylation, phosphorylation of histones to take the histones and disassociate from the DNA. That's going to be the first initial step. It's opening up the superstructures on the chromosome. And this key, again, is usually done through some sort of post-translational modifications, methylation, acetylation, deacetylation of lysine residues, all kinds of processes like that, which is a topic of an important discussion, a new emerging field in molecular biology called epigenetics. We're going to not focus on that in this lecture, but just know that that's the precursor for this process. So step zero, again, we're going to end up with relaxation of our higher order structures. One of the important enzymes is topoisomerases, and they're also called gyrases, and they temporarily introduce single or double-stranded breaks of the DNA to relax the supercoil. DNA, as you might recall, is in a helical structure, and gyrases or topoisomerases come in and they nick the DNA, and that allows them to start initiating at a replication fork. The replication of DNA molecules starts with the unwinding of that DNA double helix, and that can occur either at the end or in the middle of the DNA. And special unwinding proteins called helicases attach themselves to the DNA strand and cause the separation of the double helix. So the double helix will start, the gyrases nick the DNA and allow it to be unwind. Helicases come in and they cause the separation of the DNA. So from here, you're going to have primases. They're going to come in, they're going to bind, and they're going to start to initiate, which is most people consider step number one of the replication process. So primases initiate the complex. DNA polymerases come in. We all know DNA polymerases function 5' to 3'. So all activities added to the 3' end of DNA, which we'll show you here in a little more detail a little later. And you end up adding on DNA. The polymerases ended up adding DNA in the 5' to 3' direction. There is also primers, short RNA primers that are initiated to start replication. So, once things get going, the polymerase is going to go through and it's going to add in the DNA nucleotides one at a time, which we'll talk about in detail. Initially, though, we have an RNA primer, and there are short 4 to 15 nucleotides that prime this initiation step. So the DNA polymerase will use these to get things rolling. So once we get the complex built and everything's primed and ready to go, we're going to go into the elongation step, which is the second step of DNA replication. DNA replication is semi-conservative, meaning that each strand of the DNA acts as a template for the synthesis of a new complementary strand. So you're going to take 2 templates, you're going to get 4, and you end up with a parent and 2 parents and 2 daughter strands. So that allows for the doubling, the replication of the DNA. DNA polymerases are responsible for synthesizing the DNA. They just add nucleotides one at a time. And what you can see here is, as we're going through, you can take an ATP and it binds, A binds with T. It's going to come in and it's going to bind. Actually, in this situation, it's going to be T binding to A. TTP comes in, and two phosphates exit. We get a hybridization between the A and the T, and there's a phosphodiester bond between the two. That should be a review for most of you. Some key features of DNA polymerase. There always needs to be a template. It can't do things de novo. They only add DNA at the 3' end. They can't start making DNA chain from scratch. They have to have those primers, as we mentioned in the initiation step. They do proofread of their work, and the vast majority of the additions are correct, but they can remove the incorrect nucleotides. Most nucleotides are actually most nucleotides that are added incorrectly are repaired by the polymerase. This does require energy, and again, the energy comes from the high energy nucleotide where we have 3 phosphate groups. So in this situation, it would be TTP, we can have ATP, CTP, and GTP. So at this point, we're going to end up with ligation coming. We're also going to have ligation option, which we'll talk about here in a minute as we get into the step 2 of DNA replication. Let's talk a little bit about how this works, the fundamental chemistry. And so you start with as you might recall, you're going to start with a nucleotide, and the nucleotide, as you might recall from the carbohydrate section, has a hydroxyl in the 3 position. So that's where it's going to add, and what we end up having is the high energy nucleotide that has 3 phosphate groups. You have a nucleophilic attack on that hydroxyl group. So once you have that nucleophilic attack, you lose 2 phosphate groups, and you end up with a newly formed phosphodiester bond. Guess what? We have another hydroxyl group. Once we add that other one, that hydroxyl group again can form is a spot where we can polymerize the next base into the sequence. So that's pretty much how that's the biochemistry, chemical reaction behind replication. So again, in elongation, we have a replication fork that is formed. It's created by helicases, again, that break the hydrogen bonds together, and we end up with 2 branches. There's a leading strand and a lagging strand, and these 2 serve as templates for replication. As we mentioned, DNA is always synthesized 5' to 3', and since the leading strand and lagging strands have different orientations, we kind of have a major issue because we have one strand who's the opposite direction of the growing replication fork. So what we have to do, well, let's just start with the leading strand. The leading strand is the normal strand, and that's being synthesized the same direction as the growing replication fork. And so the polymerase just reads the template strand and adds nucleotides one by one. That's the leading strand. The lagging strand, however, we're going the opposite direction of the replication fork because it's going, instead of going 5' to 3', it's going 3' to 5'. And so we have to add these in short fragments, these fragments of DNA. And this is called an Okazaki fragment. And it's short sequences that are added in to the DNA. The RNA polymerase adds those things in. So once we get all these put together, we got 2 steps. We got to remove the primer once we terminate. We have to remove the primers and ligate everything together and make sure it's all. And proofreading, while it happens in line, can also occur later. So what we end up having in termination is we end up ending the replication fork, and we have to go back in, and we have to, the DNA ligase comes in, there's nicks in the phosphodiester backbone, it comes in and repairs those nicks and removes the RNA. That's at the termination step. So 3 steps: initiation, elongation, termination. Termination steps includes removing the RNA primer and ligase going through and taking care of all the nicks. Eukaryotes have much more DNA than bacteria, and so we have eukaryotes have multiple origins of replication complex. They're called ORCs. Each one has a replication fork, and since we have 30,000 genes and replicons, we can use these multiple ORCs to provide multiple places where the polymerases can initiate replication. We have also repair that can occur, as we mentioned earlier. When we do this process is not perfect. There are some errors. They arise in replication defects. When you have them, these can include things like the wrong bases. You have cross-linking. You have double and single-strand breaks, and this can happen during replication or after replication. And many of these errors are just detected and repaired either by polymerases or by proteins that come in, and exonucleases that'll come in and remove a base pair and repair it. This would be like what we call proofreading errors when we have a DNA nucleotide that doesn't match its complementary. We can also have insertions, deletions, and breaks, and all of these things. Special polymerases come in and can take care of that. All right, there are numerous polymerases that exist. Prokaryotes have 3, Pol1, Pol2, and Pol3. Pol3 is the primary in bacteria. In prokaryotes, Pol3 is the primary enzyme of DNA synthesis. We have, in eukaryotes, we have DNA polymerase alpha. It synthesizes the major part of the initiator polymerase. And we have alpha, beta, gamma. DNA polymerase gamma is the primary enzyme of DNA synthesis. This kind of gives you a chart of what the different polymerases are. We're going to introduce the idea of recombination. 2 DNA molecules can recombine with each other to form a new DNA molecule and have a new sequence. I know that we're going to spend a little bit more time on this, but allows genetic information between 2 DNA molecules to be spread. So 2 daughter molecules are formed as 2 parent strands of DNA come together. And it's important in this replication is very, very important in multiple systems, including things like viral genome integration. We use it in biotechnology applications. We can use it to generate knockout mice and other applications. So this is a very important process, and I'm sure that we will discuss that further. Appreciate you listening to this lecture on replication, and I look forward to seeing you in the next one.